peptides are quietly regulating nearly every system in the human body. This presentation is titled Peptide Biology, Therapeutic Classes and Translational Systems Map, presented by Brian Geniuson, research scientist. This discussion examines peptides through a structured systems biology framework, integrating molecular signaling pathways, physiological systems, and clinical relevance. Peptides are short chains of amino acids derived from proteins and function primarily as signaling molecules that coordinate communication between cells. They can be broadly classified into endogenous peptides produced within the body, natural peptides derived from external sources, synthetic peptides created in laboratory environments, and engineered peptides designed for targeted therapeutic applications. At the molecular level, peptide activity is governed by structure-function relationships, including amino acid sequence, charge, and conformational behavior. These features determine how peptides interact with biological targets. Peptides act through multiple receptor and target classes, including G-protein-coupled receptors, ion channels, receptor tyrosine kinases, immune receptors, and intracellular signaling systems. Through these interactions, peptides regulate key signaling pathways, including cyclic AMP-MAPK signaling, PI3K-AXCT pathways, NF-kappa B signaling, and JAK-STAT pathways. These pathways control metabolism, inflammation, cellular growth, and gene expression. From a systems perspective, peptides exhibit broad biodistribution. They influence the central nervous system, cardiovascular function, metabolic regulation, immune response, musculoskeletal systems, and gastrointestinal and hepatic processes. Clinically, peptides are increasingly used in areas such as metabolic disease, endocrine regulation, oncology, and emerging therapeutic applications. However, peptides also face translational challenges, including rapid degradation, limited bioavailability, and delivery constraints. Engineering strategies such as cyclization, lipidation, and advanced delivery systems are actively being developed to overcome these limitations. Key points. Peptides are short amino acid chains that function as critical signaling molecules. They regulate major biological pathways through receptor-mediated interactions. They influence multiple physiological systems simultaneously. They are increasingly important in therapeutic development. Their clinical use is expanding but constrained by stability and delivery challenges. Highlights. Peptides act through core signaling pathways, such as CAMP, MAPK, PI3K-ACT, NF-kappa B, and JAK-STAT. They demonstrate system-wide biological influence across neurological, metabolic, immune, and cardiovascular domains. They provide high specificity compared to traditional small molecule therapeutics. They represent a rapidly expanding frontier in modern medicine. From an evidence perspective, peptide science demonstrates strong mechanistic foundations, extensive preclinical validation, and a growing body of clinical research. Translational readiness continues to improve as delivery technologies and molecular engineering advance. Conclusion. Peptides represent a critical interface between molecular biology and clinical medicine. They connect cellular signaling mechanisms with system-wide physiological outcomes and therapeutic applications. As research continues to evolve, peptides are expected to play an increasingly central role in precision medicine and targeted therapeutic design. Full scientific literature supporting these mechanisms and applications is available in extended research materials. This content is presented for educational and scientific purposes only and is not intended as medical advice.